This is our fifth example of using virtual reality for data analysis. And like the title says, it's about local regression. And in virtual reality, we present a library of samples with reference values of the analyte, let's say concentration, that we want to predict. And the user identifies based off appearances of the library and the prediction sample which library samples are matrix matched to the calibration set. And I'll have a slide that quickly goes over matrix match as well as a couple slides on local regression. But first I want to explain why we're using virtual reality for data analysis. In virtual reality, we're actually looking at a fusion of all three human senses, sight, touch, and hearing. And by using these senses, the decisions are made. So we go by what data looks like, what it feels like, and the data sounds like. And humans have had a lifetime of training, so we don't really have to train the users to do the data analysis. Already come pre-trained. Whereas a machine learning algorithm takes extensive training, and depending on the data set, what dictates the type of training that has to go on. Whereas in virtual reality, it doesn't matter what the data set is, the humans are already pre-trained to do the data analysis. And by using this hybrid combination, we think we improve on just using only machine learning. And we have shown that in our other examples where we succeed, where automated algorithms fail. Like in our first example that we did was model selection. And so we were able to select better models to predict our analyte than machine learning algorithm could select models for. We did outlier detection, and the nice thing about the outlier detection is that we could identify masking and swamping, which we needed to be able to do because that was going to be a precursor actually to our third project, one class classification. And you can go to these YouTube videos as well as these papers to read more about them. Interestingly, in the one class classification, again, we were able to reduce the false positives and false negatives that machine learning algorithms were generating, as well as we were able to identify these local clusters. And for us, these were our matrix matched samples. And that would be our precursor for the current project, this fifth one, of doing model or local regression. Our fifth project, which is kind of interesting, we took blind people with some with no science background, and we kind of explained how the data is going to feel and sound. And they were doing one class classification and also reducing the number of false positives and false negatives just based off how the data felt and uh, uh, sounded. All right. So before I explain what local regression is, briefly what a calibration is all about, calibration and prediction. And so just like any kind of training set and prediction, we call it calibration in analytical chemistry. I have a vector of my analyte reference values. Like I said, that can be concentration. And for us, we usually use spectra like near IR spectra, NMR spectra, fluorescence spectra. And so we have our collection of spectra that recorded on each of these samples with known reference values. We get some kind of regression model, and with this regression model, we can then interproduct that with a spectrum of a new target sample, and we get the estimated analyte. And that all works fine, but the problem is that very often our new target samples, they're not matrix matched to the calibration set. There's some slight differences due to physicochemical properties, the molecular interactions that are going on in the sample, or there's a different secondary analyte correlation. And like, for example, for agriculture products, it could be that I built a model to predict some agriculture product from some geographical region, but now the model won't work for another geographical region. And so we have many of these hidden variables, which we call matrix effects in analytical chemistry, and they can make your prediction inaccurate. So one solution to the fact that my new samples are not matrix matched to the calibration set is local modeling. And that's where you have a large library. So this characterization here is I have a library. At each of these pixels, I could have a spectrum measured. So I have spectra of ocean or water, sand, rocks, dirt, one kind of vegetation, another kind of vegetation, lots of different vegetation. And so I have my target sample. So if this is the sample I want to predict, then the idea is I want to mine through this library and find those samples, these pixels that are kind of matrix matched to my target sample, and build my regression model and then predict. Pictured here is a cartoon characterization of the situation. So here's my reference library with known analyte values. And let's say I have these three samples that I want to predict the analyte value of. So I have their measured x's, but I don't know their y's because that's what I want to use the model for. So first thing we do is we X match, and so we matrix match the target sample spectra to the reference, and you can see we can bin this out into three separate bins, but now we need to bin the Y's. I don't know, what my Y of my target sample is, so I have no idea where I need to be to do the regression through. And that's a tough problem to solve. So again, the idea is that for every target sample, you build a new model that is both matrix matched in terms of the x's and the y's. It is a difficult problem, but we actually spent a couple of years working on this, and we're satisfied that this is a pretty good working solution. We call it local adaptive fusion regression. And you can go to the paper and read about it. In that paper, we did a couple different data sets. This is the biggest one, was this soil data set with nearly 100,000 library samples, and we randomly just pulled 50 target samples, and then we ran each one through our Laffer algorithm. And you can see, the green is the true analyte amount, and you can see the box plots of our training sets, our calibration sets for each individual sample. Does a pretty good job of closely bracketing the analyte value, even though we don't know what the analyte value in the target sample is. The thing about Laffer is it does use hundreds of similarity measures and an abundant number of filters and checks, and we run through all possible combinations of this relative to their hyperparameter values. So it can take some time. So we wanted to try and do this in virtual reality. In virtual reality, we're just going to visualize the data and visualize the matrix effects. So to do that, we need something called a glyph, and that's what we call our samples in virtual reality. And there are abstract glyphs and metaphorical glyphs. And in reality, the success of your data analysis kind of boils down to your ability to have a glyph that characterizes your data in a good way. And so there's lots of different ways to adjust your glyphs. Here's some abstract glyphs. Our glyphs that you'll see in the video is kind of like this. It's just a ball, but we have spikes all throughout the whole ball. And we also have a new glyph we're working with stacked cylinders, as I'll show you a picture of. But there's a lot of different features you can change on the glyph, and each of these features you can attribute to some sample measurement. We're also just now starting to work with some metaphorical glyphs so these are fish, and so this is for a classification of fish, and we're classifying relative to the species, whether it's fish, fresh, thawed, or frozen, and location and year of the fish. So the main class is all of these blue fish. They look like fish, and you can see if you're not matrix matched to the class, and you're not one of those varieties, then you'll not look like the same fish. And again, that's just the recognition. That's all one has to do, is to identify whether a sample is in a class or not. Is does it look like a fish or not like the fish? So to do our matrix matching, we need a lot of similarity measures of our sample, both in terms of the spectra and in terms of Y values. And we created this algorithm called PRISM, and we've used it for a lot of different uh, uh, topics, and as listed here. You can go to the paper and read about it, but basically we do create hundreds of sample-wise prism values, as we call them, similarity measures. And we have two types. We have prism X, which are just based off the measured values on your samples for us, spectral measures. So this is how we matrix match the spectra. And then we have prism Y. These are based on estimated analyte amounts. And we need this for quantitative purposes, which is what we want to do because we want to be able to predict concentration. And actually, we figured out how we combine these prism X and prism Y values into the respective prism X and prism Y values into respective prism Z scores. So the basic flow is we have sample measurements. We convert these to our prism information, similarity measures, information streams. We attach these hundreds of prism values to glyph features, whatever the glyph is, create the glyph based off the prism values. And then we're using our human senses as the new information stream to make the decisions. So these are our glyphs that you'll see in the video. Like I said, they're balls and spikes, and here are the prism Y glyphs and the prism X glyphs. And then we create a composite prism glyph. That's a composition of both prism X and prism Y. So the spikes are all our prism similarity values. We have more prism X values than prism Y values, so they'll see more spikes. The sizes are relative to the respective z-scores. We also use conformal prediction p-values, and we can adjust the sizes relative to those. And then we just went ahead and have the prism z values in there twice relative to size, and then we broke them up into 3 unique bins. And so we can immediately see whether they're small, mid-range, or large based off the color. As you'll see in the video, you touch a glyph, you'll see the prism z-score and the p-value, and as well as if it's a calibration sample, you'll get the analyte amount. And in the lower right are our new prism, or our new glyphs that we're using. Like I said, they're stacked cylinders, and what we do is it's hard to see the green balls or the green cylinder there. The cylinders are kind of neat. They're ordered from largest to smallest, so it's a conical shape for the prediction sample. And then whatever that order is of the prism values, we order the calibration samples the same way. So you can see on the far right of the lower picture, those cylinders are not arranged in a conical shape, and so those would not be matrix. Those samples wouldn't be matrix matched to the target sample. So as you'll see in the video, these are the before. We have all sorts of sizes of our glyphs, and nothing that looks, and some that look like the target sample, some that don't. And the idea is in the after is we've weeded out all of the unnecessary samples, and we're left with glyphs that are similar in size and shape and look like the green target samples. We do create these user interface boards in the virtual reality, so you can control kind of what we see. I think later in the video, you'll see we can control, we're doing principal component as our axes. We've done work with UMAP as well. And so we can control which PC to use for our axes by these slide bars. We have some scaling aspects in there. And then for this sample selection, we have a bar plot of the calibration Y values, because the idea is to shrink that Y value so we can always see what the range is and what the Y values are of all our calibration samples that are left in VR that we're going to use for the to build the model, as well as the number of samples. This is the movie showing the VR view with all the UIs. in the different prism glyphs. These are the prism Y glyphs. And you can see there, as they're being touched, you get the various z-scores, p-values, and the analyte amount, because these are all reference samples. The green is the target sample. And so the user is just looking around, isolating samples that are going to be matrix matched. They look like the target sample, and so therefore there's a good chance they're going to be matrix matched. So we're going to reduce the glyphs to this Y range of 55 to 75. And so you can see the prism Y values are okay. But now we go shortly to the prism X glyphs, and you can still see there's quite a variance there. And so we need to throw out some of the more samples and that don't match the target. So here the user is piling up some samples, and you'll see how we remove the samples, and it's easiest when they're collected into a group. So red glove takes away samples. So these are the composite, so they look pretty good. And as noted, we do iterations because the prism values will change relative to the base set of samples. So this is the second iteration. Prism values were all recalculated for the samples, and now you can see we have a variance again. So we can further reduce the number of samples and the analyte range, the concentration of water in this case, and throwing out samples. This is a quick example of showing the PC axis changing, which also changes then the cluster formation. So we're just going to do the same thing, throw out some samples in the prism Y, reducing the concentration. Then we go to the prism X and throw out samples that don't spectroscopically match. And here's the composite. And we're going to do one more iteration, a third iteration, and then we just do the same thing. And you'll shortly see that on the third iteration, we get another set of prism values with a variance, and we'll remove samples. It was after 3 iterations that it was decided that the remaining samples were about as tight as we're going to get and matrix matched. And then with that, we do our prediction, build our model and do our prediction. Like we saw before, here's our before and after. The before is the full library with a large variance in the glyphs, sizes, and shapes. And after, the goal is to have a matrix match set. So there's little differences between the ball and spike sizes. This is a more homogeneous collection of samples. And the samples are all similar shaped to the target sample. And then we build our model and predict with that final calibration set. This is how we did for this target sample that was in the video. And you can see the global, or the library, was 156 samples, quite a large range in the Y values, and our prediction error is the greatest. Where LAFER, our autonomous algorithm, and VR came up with pretty close results, indicating that VR, the human, is by visually looking at glyphs, you're able to isolate those samples that are truly matrix matched, as does the algorithm, but the algorithm has an abundance of filters that the algorithm has to march all the samples through, which takes time. In our paper we submitted, we did 5 target samples per, we did 4 different data set situations, while actually the first 3 rows here are one data set, but 3 different analytes. And you can see that we do as good or slightly better than LAFER without having to go through the extensive hyperparameter filters that LAFER has, and you just do as in the video. You just go by how it looks and you narrow it down, and we have our prediction. The VR, SB are the spiked balls that are these, and then these is done with the cylinders. So since we had three different approaches to build calibration sets for each of these 5 prediction samples per data set, we looked at the regression vectors, and you can see they're plotted here, and you can see sometimes they're quite similar, sometimes they're quite different, and that's because we have different samples. And so, but all of these models, like all 3 of the models for moisture, meat moisture, they look different, but all 3, they all predict accurately. So we got totally looking different regression vectors, and yet they all predict accurately, and that's something that's become known as the Rashomon effect in machine learning. And because of that, from our lab, we don't think you can really interpret these regression vectors, because I could come up with other regression vectors that look differently, different peaks, different places, different shape, magnitudes, and yet they all predict accurately. The last item I'll touch on are the biases and where best to use VR. Different humans are going to have different decision paths through the VR world, and just as likely the same human, but on a different day, is going to have a different path through the VR world. And these are our biases. Uh, as we've seen, the obvious matrix match samples, nobody disputes that. And the obvious non-matrix match samples, no one disputes that. The hard ones are the gray borderline situations. And to try and reduce the biases, we can use multiple glyph styles to help the user decide. Switch to a different projection view like UMAP, and to help the user decide, as well as perhaps node and links. And something we're looking at is having the user actually generate their own glyphs, so they have the biases in the glyphs, so they know what they're looking for to make their decisions. Humans do get tired. You can't do 100 samples in a row, so best when we only have a few target samples. The glyphs do allow exploratory data analysis, and that's a really powerful use of virtual reality. It allows you to correlate the sample properties, your local clusters, as well as the relationships between samples within a local cluster, as well as between local clusters again. We think computers have biases as well. That's because the user usually has to select some kind of algorithm pathway or pipeline, and there's metaparameter optimization, which are based off user inputted thresholds. And so these thresholds and pipeline choices are all incorporating human biases into the computer algorithm. Nice thing about computers, though, is they never get tired. They can just go 24/7. So if you have 1,000 samples to predict the analyte amount of, then a computer is just going to keep rolling. Where virtual reality is you're going to be bottlenecked by those number of samples in terms of turning over useful results. So thanks for listening to this YouTube, and check out the other YouTubes in our virtual reality projects.